Hello children, welcome for today's biology class. So today we are going to take the TS of stem, that is monocot. This is monocot stem, this is dicot stem. The both are fresh stems I have taken. So we are going to take TS of the stems and we are going to observe under microscope after staining with help of safranin. Safranin is the stain we are going to use here. So for this purpose the materials required are the stem cuttings, microscope, safranin, glycerin, blotting paper, cover slips, slides, brush, forceps, needles and blade for making the cutting, and water for washing the excess stem. So now let us start with the practicals. First to take a drop of water upon the slide, children. Now we can take the stem cutting. We have taken a dicot stem and we are going to make a thin slice of it in circular way without damaging. We are taking a thin slice. Can you see the thin slice? It has been taken upon the blade and this can be stained. So before staining we are keeping upon the slide. Okay, then we are going to add a drop of safranin. And the safranin stain, it has to be stained evenly. After keeping few seconds, then we can wash the stain with help of water. Okay, now few seconds over. Now we are washing the stain. Slowly we can add some water and the excess stain, it can be removed. So the cutting, it should not, the TS of the stem what we have taken, you have to handle it carefully. Otherwise it may fall inside water. Now slowly we are draining up all the water with help of brush. Okay, so now the slide, can you observe on the slide that the circular cutting what we have taken, the stained one is there. So now we are going to add glycerin, and the glycerin, it prevents from drying of the section cutting what we have taken. Okay, and now we are going to put the cover slip. So with help of index finger and thumb finger, so just slowly we are putting with using a needle. The cover slip, it has been put upon the slide. So now without air bubble you have to put upon. Now the excess stain can be removed with help of blotting paper. Now the slide, it has to be cleaned fully. Now it can be observed under microscope. This is the slide we have prepared, children. So before observing under microscope, I am showing you. So even with help of naked eye, you can observe the ring-like structure. Can you observe the vascular bundles arranged in a circular manner? So in the form of a ring-like structure, can you observe in the center? Now we are going to observe under dissecting microscope. See here, the ring-shaped structure you can observe. So in the center, you can observe the lightly stained portion. So that one, that is the pith. The pith is present in the center. This is the diagrammatic view of the dicot stem. So here you can observe the pith in the center surrounded by ring-shaped vascular bundle. So here the vascular bundle is conjoint and open. The identifying features of dicot stem are vascular bundles are wedge-shaped and are arranged in a ring-like manner. So here the vascular bundle is conjoint, means xylem towards one side, phloem towards outside, and open means containing cambium. And later the xylem is endarc. Endarc means protoxylem towards the inner side and metaxylem towards the outer side. And pith is present in the center. A large number of vascular bundles arranged in a ring-like manner with help of pith in the center. Easily we can identify the dicot stem. So here, children, once again in the microscope, can you observe in the center pith? Microscope, can you see the light colored portion, that is the pith, and the vascular bundles arranged in a ring-like manner, clearly you can observe. These are the distinguishing features of dicot stem. Thank you, children.